Hello everyone, my name is Maggie Steinhoff and I'm a third-year CMB student. Today I will be discussing my research project concerning the role of ST6L1 in xenotransplantation. Xenotransplantation is the transplantation of non-human animal products into a human patient. This is an area of active research because there are more people on the transplant organ waitlist than there are available human organs. This has left a large deficit that continues year after year. In 2019, there were approximately 112,000 waitlisted patients, and while there was a record-breaking almost 40,000 transplants, that only accounts for about 35.5% of all the waitlisted patients. This issue is further compounded by the fact that every 9 minutes, a new patient is added to the waitlist, and that every day, 17 people die while waiting for a transplant organ. In order to solve the supply problem for transplant organs, we're turning to a new source of donor organs, pigs. Pigs can be used because their organs are similar to humans in size and function, they can be genetically modified, and there is a reduced risk of zoonotic disease transmission versus primates due to the fact that humans and pigs are a little more distantly related, and the animals can be raised in specific pathogen-free or even clean facilities. Unfortunately, we can't use pig organs in people just yet. This is because there's molecules on the surface that cause hyperacute rejection in humans that can be seen in as few as 10 minutes after transplant. The removal of these major xenoantigens has been the focus of previous research and has resulted in the elimination of hyperacute rejection. But this still leaves acute and chronic rejection issues that will need to be addressed before xenotransplantation for human patients can become a reality. Before I get any further into my research project, I first want to give some background information concerning sialic acids. Sialic acids are 9-carbon sugars on the ends of glycans present on the surface of cells. They have many diverse functions, as you can see by the chart on the screen. However, the most important one for my work is the self-recognition capability of the sialic acids, as highlighted. This is the pathway that I will be exploiting for my research project. When CD22, which is a Sialic exclusively found on B cells, binds specifically to alpha-2,6 sialic acids, it prevents the activation of the B cell. This would be a step towards helping to prevent chronic rejection of a xenograft, as B cells are the immune cells responsible for the production of graft-specific antibodies, which would then lead to complement activation against the graft and potentially even adaptive immune response against it as well. Since alpha-2,6 sialic acid recognition is the major goal of my project in order to prevent immune cell recognition, I first need to increase the surface expression of those sialic acids. This is done by inserting an additional copy of the gene, which encodes for the enzyme ST6L1, which produces alpha-2,6 sialic acids. These ST6L1 transgenic cells are then used along with human PBMCs in order to predict how human immune system would respond to the cells. These human immune cells and the ST6L1 cells are co-cultured together for 6 days, and the proliferation of the immune cells is measured by flow cytometry. The proliferation percentages, shown on the bar graph, are the proliferation rates of the immune cells cultured with ST6L1 cells after comparison to the rates of immune cells cultured with wild-type pig cells. So pig cells that express transgenic ST6L1 evoke less proliferation than wild-type pig cells. That's true anyways in 2 of the donors, although donor 3 does show a very low increase in proliferation for both T cells, but they do then follow the overall trend with the reduction of B cell proliferation, which is the most important aspect for my project. This observed overall reduction of immune cell response to the modified cells has made us optimistic that inserting an additional copy of ST6L1 along with other genetic modifications will bring us closer to making xenotransplantation a reality. In the meantime, the next steps for my project are to analyze the CD22 pathway of B cells in response to the ST6L1 transgenic cells, just to ensure that we are correct that this is what is responsible for the reduced immune response. And then I want to compare the levels of sialic acids present on human and pig organs. This will just confirm that by increasing the levels on pig cells, I'm making them appear closer to human cells rather than highly overexpressing to the point that could prove detrimental to human recipients in the future. I hope you enjoyed hearing about my research and how I hope it will bring xenotransplantation closer to being a viable option for waitlisted patients. Thank you for listening.